Hi, my name is Tarah Rhoda. I am the manager of the SVA Bio Art Lab. We are a bio lab facility, Bio Level 1, and we work with artists and creators that have very little to no background in the sciences. It's a wide spectrum of accessibility in our lab. We work with a handful of biomaterials and laboratory techniques and protocols that can be applied in a creative manner. I'll show you around the space and show you some of our specimens, our miniature ecosystems, and some of the biomaterials we cultivate. So we are one of the very first biolabs in an art school in the US. Like I said, we've been open for about 5 years. We're in the midst of our summer residency, so artists from all over the world have joined us, some for their second and third residencies. This is Carolyn, who is one of our very first residents, so she's seen just how much the lab has grown. So we work with a variety of specimens, some of which have been preserved. Here you'll see some of our collections. Many of these specimens have been stained and cleared, which is a technique to render their image their skeleton. Many of them have been saturated with pigments that target their skeleton and their cartilage. And then they go through a series of digestive enzymes that literally eat the soft muscle tissue until the specimen is rendered clear. We also quite like working with live specimens as well. These are 2 of our saltwater tanks where we have a variety of soft and hard corals. Here we have some freshwater tanks. This side represents the Congo and this side the Amazon. And while this is one of our more modest tanks, it's our favorite spot in the whole lab. This is the Saltwater Hotel. And here in the corner you'll see our very special guest. This is a baby skate who hatched in captivity, which we were told was an impossible feat. So this is the Saltwater Hotel. Students can come to this tank and pluck out an urchin or a starfish and bring it over to one of the microscopic imaging setups and image it feeding and producing waste and things of that sort. One of the techniques that students frequently start with, as this can be a fairly timid space and unfamiliar territory, is microscopic imaging. So behind you you'll see some examples of our setups. We typically have a camera mounted using the lens. The lens of the camera is essentially the microscope itself, with the live feed connecting to the laptop. So students can take high quality still, time-lapse, and videos. Here we have some more of our ecosystems. We've got some grafted cacti, which are very simply 2 different cacti fused together. We're hoping to eventually make a totem pole of 12 or so different cacti species living as one fused plant. Here we have our praying mantis collection. So you'll see these are all carnivorous praying mantis, and we have our carnivorous farm as well. So these are primarily Venus flytraps, morning dews, and pitcher plants, and they eat genetically modified flightless fruit flies. So they have wings, they just don't know that they can fly, which makes feeding the masses a bit more reasonable. We've been feeding this one fingernail clippings, because the enzymes, you'll see that they all have little trip hairs, and when you make contact with the trip hair, the mouth closes and begins producing an enzyme. So you can see some examples of little miniature ecosystems that students have worked on. This is our project rack. So this is where students are keeping cultures and materials in progress. When I mentioned biomaterials earlier, I meant organisms that have some degree of image or mold making capabilities. And there are 2 that are very elementary and easy to use. And we refer to these frequently as 2D and 3D biomaterials. So here you'll see sort of a fleshy-like disc floating in this liquid, and that is a byproduct of kombucha tea. It's called a SCOBY, which stands for, it's an acronym, it stands for a symbiotic colony of bacteria and yeast. So it takes the diameter of the container it grows in and then can be dehydrated to have a leather-like quality. So it can be tattooed, sewn together, has a very performative quality. Students can literally grow their own canvases. Another biomaterial we work with is called mycoform, and it's a byproduct of the mushroom industry. So it's simply mycelia, which are the roots of a mushroom, digesting a cellulose mixture. So this plus this, within a week or so, will give you something of that sort. The mycelia is digesting the cellulose mixture and taking the shape of the container it's grown in, which is why we refer to it as a 3D biomaterial. So these are two very introductory organisms that can be applied for construction materials. And here we have some of our Mayo tomatoes behind our projector screen here, and I'll tell you a little bit more about that in a moment. Another biomaterial we work with, and this is a bit more illustrative, are bacteria paintings. Students can create living paintings. Primarily we work with a culture called E. coli K12. It's been disarmed, so it doesn't take up residence in the gut. It's not the type of E. coli you hear about in the news. And it's been genetically modified to express a protein that's naturally found in a bioluminescent jellyfish. It's called GFP, the green fluorescent protein. So students can create living paintings. And one of the really interesting things is that it only needs the nutrients and the moisture of the agar to grow, not to glow. So once the bacteria has been established, you can print it on other materials like fabric, paper, leaves, live plants, and you can put your empty dish back in the incubator and your image will regrow. So those are some examples of organisms that can be applied in a traditional art practice. And here I'll show you our plant tissue culture room. So this is where we have some of our more rigorous plant projects. The lab does host a wide spectrum of projects. Some are simply reference-based, microscopic imaging. And then we have some students that catch a lot of momentum and go off the deep end and want to solve world hunger and design their own plant. So this is a project called the Mayo tomato. Our students presented it at the Modern Museum of Art last summer. They worked alongside a DIY biohacker named Sebastian Choba and created what's called the Mayo tomato. They wanted to combat the idea that the future of protein was lab-grown meat, which is more sustainable but still requires a lot of resources. They wanted to create a scenario where the average Joe could grow his own protein source on a fire escape in Bushwick. So this is the Mayo tomato, a tomato that produces myoglobin, a protein found in red meat. So theoretically, each tomato would have the protein equivalent of a burger or steak. And these here are the actual Mayo tomatoes. The students themselves were more concerned with creating a narrative around such a project, considering the speculative design and the social implications. But in the background, they were working with a biohacker to actually try to genetically modify the thing, which was pretty exciting. Here you have some African violets, and these were our very first successful crop of plant tissue culture, which is essentially cloning plants. So we were able to grow all of these African violets from a small piece of tissue, this big. And this is a technique called plant tissue culture, which basically yanks the plant back to scratch. It grows in these sort of callous tumor-like clusters of unorganized cells, typically in a very clean vessel. And then we introduce hormones that are naturally found in nature that give the plant instructions to develop its roots and its shoots. Once the plant has matured, we can acclimate it to the harsh, cold world, and hopefully it matures and fruits for us. And behind, you'll see some of our incubators. So students have, most of these are mycelium cultures. So you can see the students are experimenting growing the mycelia on different substrates. So we have coffee grounds and some cardboard in this vessel. Yes. But other than that, the lab tends to tackle a variety of projects. We work with a lot of food science. We do a lot of DNA testing. The students in our summer residency program are about to follow up on their mitochondrial DNA results, so they'll learn about their mother's heredity and their haplogroups. So if you want, we can take a peek at the molecular biology bench. I think we have Ellen stationed here preparing for the workshop. So this is where all the magic happens. This is where all the media is made for the bacteria paintings. This is where the DNA is processed, and the bulk of the more technical work takes place.